inherited dogs deficient in panties by early one-sex invasive infection. Definition: any defects that cause combination immunodeficiency by correct susceptible to early one-sex severe infections by a variety of virus, bacteria, fungi, and parasites. Autoimmune manifestation, allergies, and cancers can also occur.
Early severe cases with multi-system disorder affecting multiple tissues. Through the efforts we've made at identifying genes that cause this disease, we now believe that the underlying basis is a defect in telomere maintenance. So this study really begins with a family from Birmingham, referred to as two very young children, presenting age of 3 and 5 years, with classic features of the disease, mucocutaneous features, bone marrow failure. Interestingly, neurological abnormalities is cerebellar hyperplasia, and the hallmark of the disease.
Characteristic by early one-sex invasive bacteria and viral infections, parasites, T cells, B cells, and NK cell function. Definition: differentiation immunity in both hematopoietic and non-hematopoietic cells.
Natural killer cells kill on contact. The killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are
Treatments. With treatments wise recombinant interferons and alpha V and leverages mediante various types due to expression protects the definition fibroblastic prox virus-induced cell death.
Data will then go out to these uninfected cells, all these ones over here too, and what it'll do is it'll induce gene expression to make AVP. Now what AVP is it's antiviral proteins. So they'll basically start making proteins to help fend off from the virus when it is released from the cell. So the AVP will basically decrease the viral replication, and it'll help these surrounding cells to have basically less virus go out to them. So eventually this AVP that's been induced by the interferons will slow down viral replication to hopefully end viral.
Pros and cons. Not difficult to diagnose, exit to try in early detection, could lead to other fatal disease, could be fatal of disease diagnostics unlethal.
To produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
Conclusion. Autosomal recessive type 2 deficiency is a new medical disorder with complex defects of hematopoietic and hematopoietic immune children with clinical features combines immunodeficiency, especially
A child will inherit one copy of the usual gene and one copy of the altered gene, and so will be a carrier for the condition. There is a 1 in 4 or 25% chance that a child will inherit a copy of the altered gene from each parent and so inherit the condition. As each one of the 4 possible outcomes shown here is equally likely, in each pregnancy there is a 1 in 4 or 25% chance that a child will inherit the autosomal recessive condition. It can sometimes be important to explain to parents that as chance has no memory, the 1 in 4 probability of inheriting 2 copies of the altered gene for the condition applies each time 2 parents who are carriers decide to have a child, irrespective of how many children they may already have who have or do not have the condition.